These are centrifuge tubes. For this experiment, we will label one with plus pGLO and the other with minus pGLO. Each tube should be labeled with your group's name. Place the tubes into the foam tube rack. Obtain a sterile 1 ml pipette. Open it. This graduation represents 250 microliters on the pipette. Open each tube and transfer 250 microliters of transformation solution into the tubes. Place both tubes on ice. Now take the E. coli HB101 starter plate and use a sterile loop to pick up a single colony. Immerse the colony into the transformation solution at the bottom of the tube, plus pGLO. Spin the loop in the solution until the entire colony is dispersed. Repeat the step using the minus pGLO tube. Now immerse a sterile loop into the plasmid DNA tube. Withdraw a loopful of the plasmid. Ensure there's a film of plasmid across the loop, similar to seeing a soapy film across the ring when blowing bubbles. Mix this loopful into the tube labeled plus pGLO. The plasmid will not be added to the minus pGLO tube. Next, we will push the tube down into the foam rack and incubate them on ice for 10 minutes. While the tubes are incubating, we will label the bottom of our auger plates. Tubes have now incubated for 10 minutes. Place both tubes into a 42°C water bath for exactly 50 seconds. After 50 seconds, put them back on ice for 2 more minutes. Two minutes are now up. Place the tubes on the bench top. Using a new disposable pipette, transfer 250 microliters of LB broth into the tube labeled plus pGLO. Using a new disposable pipette, transfer 250 microliters of LB broth into the tube labeled minus pGLO. Now incubate these tubes at room temperature for 10 minutes. After the 10 minutes, we will now mix the tubes by flicking them with your fingers. Then obtain a new sterile pipette. Here's the graduation for 100 microliters on the pipette. We will transfer 100 microliters of the transformed plus pGLO and controlled minus pGLO suspensions into the corresponding plates. Now, using a new sterile loop, spread the suspensions evenly onto the agar surface by spreading the bacteria in all directions and rotating the plate a quarter turn between spreads. Use a new sterile loop for each plate and repeat the step with the remaining plates. Lastly, we will stack the plates and tape them together. Ensure the group name is listed on the tape. Now place the plates upside down in a 37 degrees centigrade incubator overnight. For the last part of the pGLO experiment, we will look at the plates under a UV light.